microbes in household products. Microbiology is the scientific study of microscopic organisms and their activities. They, based on their form, structure, metabolism, growth, reproduction, and identification of microorganisms. Microorganisms are microscopic living organisms which may be single-celled or multicellular. Microorganisms was discovered in 1674 by Antony van Leeuwenhoek using a microscope of his own design. Microbes. Microbes are minute unicellular organisms that are invisible to the naked eye. They are microscopic forms of life include bacteria, fungi, protozoa, algae, and viruses. Viruses are infectious agents. They are not cellular organisms. The term microbe is taken from the French and means a microscopic organism or microorganism. Microbes are present everywhere in soil, water, air, inside our bodies, and that of animals and plants. A microorganism that harms its host is also called a pathogen. Microorganisms differ widely in form and life cycle. Microorganisms live in water, soil, skin, atmosphere, floors, walls, digestive tracts of animals, humans, etc. Microorganisms play an important and dominant role in all fields of human endeavor, industry, agriculture, problems connected with food, shelter, and clothing, and in the conservation of human and health and combating diseases. Distribution of microorganisms. Microorganisms are exploited in biotechnology, both in traditional food and beverage preparation and in modern technologies based on genetic engineering. The microbial population in our environment is both large and complex. Microorganisms are used in brewing, wine making, baking, pickling, and other food making processes. They also used to control the fermentation process in the production of cultured dairy products such as curd, cheese, butter, buttermilk, and so on. The cultures also provide flavor, aroma, and inhibit undesirable organisms. They carry out different types of fermentation. Fermentation. Fermentation is a metabolic process that produces chemical changes in organic substrates through the action of enzymes. Extraction of energy from carbohydrate in the absence of oxygen. Main function: to convert NADH, a chemical compound found in living cells, back into the coenzyme NAD+. It can be used again. This process, known as glycolysis, breaks down glucose from enzymes, releasing energy. Fermentation contains two types: lactic acid fermentation, alcoholic fermentation. Microbes play a major role in fermenting food items. During food fermentation, microorganisms produce enzymes to break down complex compounds to simple biomolecules for several biological activities. Proteinase, amylase, mannase, cellulase, and catalase in many fermented foods by Bacillus species. Among these, bacteria isolated from fermented foods. Microbes are commonly present in food items like curd, cheese, bread, duff, wine, toddy. Microbes are essential in processes like wine making and cheese making, fermented beverages. Bacteria called lactic acid bacteria grow in milk to convert it into curd. The common microbes in household products: Saccharomyces cerevisiae, Lactobacillus acidophilus, Lactobacillus bulgaricus, Lactobacillus lactis, Streptococcus thermophilus, Propionibacterium shermani, Leuconostoc mesenteroids, Spirulina.
Curd. Curd is a dairy product obtained by coagulating milk in a process called curdling. Lactobacillus converts sugars into lactic acid by means of fermentation. Milk contains a sugar called lactose. When pasteurized milk is heated to a temperature of about 30 to 40 degrees Celsius or even at room temperature, a small amount of old curd or whey is added to it. Lactobacillus in curd starts to grow. This converts lactose sugar to lactic acids. It causes coagulation and partial digestion of milk protein casein. The curd forms because of the chemical reaction between the lactic acid and casein. During fermentation, the bacteria use enzymes to produce energy from lactose. The byproduct of ATP production is lactic acid. Lactic acid acts on the globular proteins present in the milk and denatures them. Lactose in milk is converted into lactic acid by several probiotic microorganisms. Fermentation depends on the temperature and humidity. It contains a number of organic acids and vitamins including B12. Lab checks growth of disease in stomach and other parts of digestive tract.
Yogurt. Yogurt is produced by curdling milk with the help of Streptococcus thermophilus and Lactobacillus bulgaricus. Temperature 45 degrees Celsius for 4 hours. Flavor of lactic acid and acetaldehyde. Made by fermenting milk with a yogurt culture. High in protein, calcium, vitamins and live culture or probiotics which can enhance the gut microbiota. Less lactose than milk because the lactose is used up in the fermentation process. Buttermilk. Buttermilk is a fermented dairy drink. The liquid left over for churning butter from cultured or fermented cream. Naturally occurring lactic acid producing bacteria in the milk fermented it. Acidulated product formed by inoculating skimmed milk with started culture of Streptococcus gramoris, Streptococcus lactis, Lactobacillus acidophilus, Leuconostoc species at 22 degrees Celsius for 18 hours. Acidulated liquid left after churning of butter from curd is also called buttermilk. The common microbes in household products: Saccharomyces cerevisiae, Lactobacillus acidophilus, Lactobacillus bulgaricus, Lactobacillus lactis, Streptococcus thermophilus, Propionibacterium shermani, Leuconostoc mesenteroids, Spirulina.
Curd. Curd is a dairy product obtained by coagulating milk in a process called curdling. Lactobacillus converts sugars into lactic acid by means of fermentation. Milk contains a sugar called lactose. When pasteurized milk is heated to a temperature of about 30 to 40 degrees Celsius or even at room temperature, a small amount of old curd or whey is added to it. Lactobacillus in curd starts to grow. This converts lactose sugar to lactic acids. It causes coagulation and partial digestion of milk protein casein. The curd forms because of the chemical reaction between the lactic acid and casein. During fermentation, the bacteria use enzymes to produce energy from lactose. The byproduct of ATP production is lactic acid. Lactic acid acts on the globular proteins present in the milk and denatures them. Lactose in milk is converted into lactic acid by several probiotic microorganisms. Fermentation depends on the temperature and humidity. It contains a number of organic acids and vitamins including B12. Lab checks growth of disease in stomach and other parts of digestive tract.
Yogurt. Yogurt is produced by curdling milk with the help of Streptococcus thermophilus and Lactobacillus bulgaricus. Temperature 45 degrees Celsius for 4 hours. Flavor of lactic acid and acetaldehyde. Made by fermenting milk with a yogurt culture. High in protein, calcium, vitamins and live culture or probiotics which can enhance the gut microbiota. Less lactose than milk because the lactose is used up in the fermentation process. Buttermilk. Buttermilk is a fermented dairy drink. The liquid left over for churning butter from cultured or fermented cream. Naturally occurring lactic acid producing bacteria in the milk fermented it. Acidulated product formed by inoculating skimmed milk with started culture of Streptococcus gramoris, Streptococcus lactis, Lactobacillus acidophilus, Leuconostoc species at 22 degrees Celsius for 18 hours. Acidulated liquid left after churning of butter from curd is also called buttermilk. Cheese. Most cheeses are made with starter bacteria from the Lactococcus, Lactobacillus or Streptococcus genera. Swiss starter cultures also include Propionibacter shermani, which produces carbon dioxide gas bubbles during aging, giving Swiss cheese or Emmental its halls. Different varieties of cheese are known by their characteristic texture, flavor and taste, the specificity coming from the microbes used. The large holes in Swiss cheese are due to production of a large amount of carbon dioxide by a bacterium, Propionibacterium shermani. The Roquefort cheese are ripened by growing a specific fungi on them, which gives them a particular flavor. Bread. Selected strains of baker's yeast Saccharomyces cerevisiae are grown on molasses. When sufficient growth has occurred, baker's yeast is harvested and converted into either powder or cakes. The same is needed. The kneaded flour is kept at a warm temperature for a few hours until it swells up. The phenomenon is called leavening. Leavening is caused by secretion of 3 types of enzymes by yeast. If the temperature is too high, for example during the cooking process, the yeast is killed. During fermentation, carbon dioxide is produced and trapped as tiny pockets of air within the dough. This causes it to rise. During baking, the carbon dioxide expands and causes the bread to rise further. The alcohol produced during fermentation evaporates during the bread baking process. Bread looks puff appearance. Dosha, upma, idli. Fermentation makes food light on tummy and make food easily digestible. Fermented preparation of rice and black gram. The two are allowed to ferment for 3 to 12 hours with airborne Leuconostoc and Streptococcus species of bacteria. Carbon dioxide produced during fermentation causes puffing up of the dough. Acidification. In this process, the lactic acid is produced. Fermentation improves nutritional value and digestible protein of rice and gram dal. Heat allows the growth of bacteria, yeast, which along with availability of oxygen and sugar decompose into carbon dioxide and water. The role of lactic acid bacteria is to reduce pH of batter to level 4.4 to 4.5, and this pH is optimum for yeast activity. Overnight fermentation of idli batter allows the rapid growth of naturally occurring microorganisms, Leuconostoc mesenteroids and Streptococcus thermophilus in grains or legumes or utensils. These microorganisms divide and produce lactic acid and carbon dioxide that make the batter anaerobic and leaven the product. Bubbles of carbon dioxide form a froth on top of the fermentation mixture. Mushrooms. Fungus like mushrooms and truffles are directly used as food. It is sugar-free, fat-free and low calorie value. It is rich in proteins, vitamins, minerals and amino acids. Cultivation is the technique to develop the fruit bodies of edible fungi. Some common edible mushrooms are white button mushroom, Agaricus bisporus, paddy straw mushroom, Wolveriella wolvacea, oyster mushroom, Pleurotus florida. Mushrooms are the fruiting bodies of a fungus, the fungus also called Mycelium. Mushrooms are produced with the help of spawning. It is good to produce new buds of mushrooms. It is cultivated in dark room with the help of paddy straw and spawn. Paddy straw helps in moisturize the spawn to grow in dark medium. Preparation of compost of mycelium is most important. Opening up the straw so that the straw absorbs water and the mushroom fungus, the mycelium, can grow in the straw. After this phase, the compost is called phase 1 compost, fresh compost. This includes phase 2. Spawn consists of specially prepared grains of corn which grows on with the mycelium. From the spawn, the mycelium grows through the compost. This kombucha, made by fermenting a sweetened tea, symbiotic culture of yeast species and acetic acid. Formation of a cellulosic pellicle layer floating on the surface of the growth medium. Composition of yeast and bacteria in the kombucha symbiotic consortium is highly variable. The pH decreased from 3.9 to 2.9. The acetic acid bacteria and yeast counts in the broth increased up to 6 days of fermentation. Acetobacter xylenum, Acetobacter aceti, Acetobacter pastorianus and Gluconobacter oxidans. Here, yeast to produce ethanol, and the presence of ethanol facilitates the growth of bacteria. Acetic acid bacteria convert glucose to gluconic acid and fructose into acetic acid. This results in formation of fermented drink. Toddy. Palm wine or toddy is served as either nira or petanir. The sap is extracted and collected at taper. Typically, the sap is collected from the cut flower of the palm tree. A container is fastened to the flower stem to collect the sap. The white liquid that initially collects tends to be very sweet and non-alcoholic before it is fermented. Palm sap begins fermenting immediately after collection due to natural yeast in the air. Within 2 hours, fermentation yields an aromatic wine of up to 4% alcohol content, mildly intoxicating and sweet. Longer fermentation produces vinegar instead of stronger wine. Microorganisms that are responsible in fermenting wild date palm, Phoenix sylvestris sap into wine were isolated and identified. Saccharomyces cerevisiae, Schizosaccharomyces pombe and some more bacteria were encountered in the freshly tapped sap. A majority of these microorganisms were also isolated from the traditionally fermented fresh toddy samples. In a fresh sap and fresh toddy, certain variations in the growth pattern of these microorganisms were noticed. In addition to this, the amount of ethanol, volatile acid, non-volatile acid, and esters produced during these fermentations also varied. It is obtained millet rice and all rice. Millets and all rice are becoming increasingly popular among health conscious owing to their immense health benefits. Considered as one of the best foods that can provide nutrition and health to your body. High energy food which is packed with iron, rich in fiber, protein, minerals such as magnesium, zinc, manganese, folic acid, amino acids, lecithin, potassium, B-complex vitamins and calcium. Niacin in B-complex vitamins reduces cholesterol. Phosphorus plays an important part in the structure of body cells and magnesium essential for our health. Microorganisms may be indigenous to the food or may be added as a starter culture after pre-treating or cooking the product. The use of lactic acid bacteria increases the acidity and decreases the pH of the substrate, thereby inhibiting many pathogens. The fermentation process increases the nutritional value of rice. Soaking rice overnight for around 12 hours increases the iron content by 21 times compared to regular cooked. This contains more amount of probiotics which helpful for digestive tract and gut. Lactobacillus rhamnosus, Bacillus subtilis are common examples present in fermented enzymes. Enzymes are proteinaceous substances of biological origin which are capable of catalyzing biochemical reactions. Enzymes are substrate specific and carry out a specific catalytic action. They work best at room temperature and near neutral pH with the exception of several digestive enzymes. Use of enzymes. Technique of immobilization of enzymes inside artificial cells or gels. Most of them are obtained from microbes. Lactases. They are obtained from Saccharomyces fragilis and Torula cremoris. The enzyme convert lactose into lactic acid. Lactic acid can coagulate milk protein casein. Lactases prevent crystals formation, that is sandiness, in dairy preparations like ice cream and processed cheese. Cellulases. Cellulase is any of several enzymes produced chiefly by fungi, bacteria, and protozoans that catalyze cellulolysis, the decomposition of cellulose and of some related polysaccharides. Cellulase is used for commercial food processing in coffee. In many bacteria, cellulase in vivo are complex enzyme structures organized in supramolecular complexes, the cellulosomes. They can contain. Conclusion. Microbes are a very important component of life on Earth. Many microbes are very useful to human beings. Not all microbes are harmful to pathogens. We are using microbes and microbially derived products almost every day. Many microbes are used commercial and industrial production of chemicals, enzymes, and bioactive molecules. Human beings have put microbes to that they play an important role in human welfare.